There's quite a bit of information in the handout, and I just took two different areas to kind of discuss with you today. Again, I've been privileged to be able to work with a number of really excellent ruminant nutritionists and reproductive physiologists over the years that were listed on that first slide, and it's been an exciting area, and I just want to kind of go through these two areas a little bit about the transition period and body condition loss, and a little bit on methionine. In our dairies, obviously we have a lot of differences out there, but our dairy cattle reproduction in general, if you've been watching it, is really improving. For many years we were decreasing, but we've been coming back, and there's a lot of reasons for that. This is a herd in Wisconsin, and you can see heat abatement here, even though we have only a few months of really hot weather. Producers are finding that to make them money. Excellent TMRs, really a lot of fantastic facilities. And in these kinds of environments, we've been having a lot of success with our reproductive management programs. And as we get reproductive management programs in place, we're able to look at some more of the specific aspects of nutritional challenges or nutritional effects and how they're affecting our reproduction. So this is one thing that is pretty common, obviously. I don't know, on our dairies we don't use it enough for a management tool, but still body condition score is very easy to do. It's a good measure of some of the amount of fat. It's used to really assess energy balance in the field. And so I'm going to use this as a measure. In general, if you're under 2.5 or the cows are under 2.5 or lower, they start to not cycle, and we start to have decreases in reproductive function. In general, the animals that are above 2.75 at the time we're breeding them are pretty much equal in fertility according to the program we're using. So we're going to look at a little bit about change in body condition score, because that's probably more critical for reproductive function and reproductive processes than actually the absolute body condition score, and particularly if we're above this 2.5 level. This is kind of the classical slide of how energy balance goes during the lactation, with energy required, energy ingested, some negative energy balance, maybe positive energy balance. And again, the negative energy balance may be having an effect on when the animal first ovulates, the risk of becoming too thin, risk of becoming too fat. You look at this time period, voluntary waiting period of 70 days, we're putting together programs that we use on a lot of our really good dairies in the Midwest and a lot of parts of the country and the world, where the animals are bred right at the end of the voluntary waiting period, and we're having over 50% of them becoming pregnant on some of our dairies. And so this time period can even get shorter for some of the animals, and they really aren't going through some of this time period here. And many times this is becoming a lot of our dairies, we're not seeing a half or one body condition score loss. They're much flatter than that on a lot of our dairies. And I'll show a little bit of that from a study we've done. But some of this, there still is the thought about the negative energy balance. I don't know if maybe our genetics are getting better or our programs are getting better, but it seems like we're not going through quite as dramatic of a negative energy balance when we're going out in the field these days as, say, 10 years ago. But still, there's some thoughts about this of how that's affecting reproduction. So this is kind of the thought of where you have calving. These first 3 weeks is where most of the change in body condition score is happening. And if you have negative energy balance, it alters the oocyte quality. So you're growing up this follicle in this negative energy balance, and then 60, 70, or whatever number of days later, when you're doing artificial insemination, that then impacts the fertility. That's kind of the thought that a lot of people have had. I have to admit to have been pretty skeptical of this idea. It's a long time period. And I kind of wondered about the exact effects independent of cyclicity. Clearly there's effects on cyclicity. And so you have this negative energy balance lowering fertility over at this point. These really comes from this idea from Jack Britt that he did at a nutrition conference, where he had these two groups of cows. Now, these were called the low body condition score cows, but what you actually see is at the time of calving, they were higher body condition score. Kind of like Rich Erdman was talking about yesterday, the animals that are higher body condition score tend to lose more body condition. So they weren't lower beforehand, they were lower in this time period, and the high body condition score animals were really just flat. They didn't really lose body condition. Not all animals lose body condition. And we're continuing to see that even in high production herds. There's about half almost of animals that don't lose body condition or lose very little. Now, they may still be losing some body fat, but as far as measurement, the subjective measure of energy balance, they aren't really losing body condition. And that was what was seen in this early study also. So these are the data put together. There were only 46 and 30 animals in these two groups, not really enough statistical power to look at reproductive responses, and yet there were reproductive responses that they saw. There was no difference in milk production, but boy, what a difference in conception rate. And that idea went out like wildfire, and you know, all across the industry, we talk about this effect on the oocyte. Like I say, I was pretty skeptical about this idea, but I was part of this trial. Rick really just brought me in. These are some data from a trial that Rick Grummer's graduate student did, and we happened to do some of the reproductive ultrasound and evaluation. And he had these 3 different dry periods. So there was a 0-day, no dry period, that's this green line. And these animals, the reason they're interesting to this talk is they had essentially no negative energy balance. They pretty much stayed flat during this time period. And then there were the other 2 groups of a 28-day dry period. They had less energy balance, or a better energy balance than the 56-day, but you can see there's definitely a negative energy balance happening there. But this group was what was interesting. And this wasn't really a trial that was powered for reproductive measures as far as conception rate, but you could see the days to ovulation. Essentially, these animals that had the short dry period, very short dry period, these are individual cows, and this is the mean, they ovulated the first follicular wave. As soon as they had a follicular wave come up, which almost all animals have at about 7 days, they ovulated it. So 13 days to first ovulation. These animals a little later, these animals a little bit later, and a lot more spread. And here, even though it isn't powered for reproduction again, we had significant effects of where the conception rate was just phenomenal in this group versus this group. And we did some other studies, but we never really did adequately this 0 days dry. We did 28 versus 56 and did have some effects on reproduction that were a pretty large study, and it did have some positive effects when we reduced the dry period. But this really got me thinking about this idea, wow, when you have no negative energy balance, maybe you can really get fertility in these animals a lot. So we decided to go back and test this, and this is a study, number of people on it. Paulo Carvalho is the lead author. You can look at it. There's 3 different experiments in this study. All of them, I think, powered correctly for statistical analysis of repro data. I'm only going to show one at this time just because of time constraints. But we were looking at this relationship between fertility and postpartum changes in body condition and how that's affecting reproduction. So here's how this study done. There were 2 dairy farms. We evaluated about 1,900 cows. They were all synchronized with double ovsync, and I'll show you why that's the case. Because we wanted to breed them at a set time, and we wanted to solve the hormonal problem. Our lactating dairy cows metabolize hormones, and if you do some of these high fertility programs, you get rid of those problems. So we were trying to get rid of kind of the hormonal metabolism problems and really look at the nutritional effects here. Body condition score was then evaluated by a single person at calving and 21 days later. Here's how the data came out then for these 3 groups. There was one group, 42% of the animals lost body condition. And most of them lost just a quarter body condition score. I'll show you that these particular dairies had good reproduction. They were managed with the Goldilocks, as we talked about, type of dry period where it's higher fiber. And so they were not high body condition when they calved, 42%. Maintained body condition, 36%. Gained body condition, amazingly enough, 22%. This is from calving to day 21. Here's the data, the exact same thing I showed you here. Lost, maintained, gained. Here's the percentage of the cows in that group that are first lactation, and there's a little bit of a difference, but there's still some first lactation, some older cows that either lose, gain, or maintain. Here's body condition score at calving, essentially 3. Again, these two herds had not high body condition. And here's, again, lost. They lost about, on average, 0.29, so most of them were 0.25. No loss and a gain. Milk production during the first 21 days, not different. There were some differences later on, but that was primarily due to the parity differences within these groups. So here's double OVSync. Maybe some of you have seen this kind of program. These are can you hear this? Okay. Yeah. This is double OVSync. It looks complicated, but it's, again, a program that's been put together over the years to be able to solve the hormonal metabolism problems. When you go out to a high production herd, if you don't solve some of those problems, most times we're going to have 30 to 35% conception rates if we're breeding to estrus and cherry picking, etc. You do a program like this, you're going to get up close to this 50% range, and so we're really happy. These are protocols that have been kind of a lifetime of various of us working with these protocols to try to get them correct for the physiology. And so this allowed us really to look at this timed AI. It was 1 week time period. We knew they were all bred at the same time under the same kinds of conditions. What effect does that postpartum body condition score loss really have in a big number of animals where we could really look at this? So again, it's higher progesterone during this follicle growth, which seems to help us. So here's how it came out. It's kind of amazing, actually. Couldn't hardly believe it. There are 400 animals in this group, 423, and like 400 of them got pregnant. The animals that were really in good, responded well to the nutritional program, so they didn't lose body condition, in fact, gained body condition, 84% conception rates, this program. If we had the other groups, they were lower. This is the 40-day pregnancy diagnosis. This is the 70-day pregnancy diagnosis. I mean, how would you like to have a whole herd of 80% conception rates at 70 days after a breeding? I mean, that would be phenomenal. Maybe we're moving that way with our genetics, etc. But this really argued that animals, if we can get animals and they're coming along, they're pretty flat, they maybe even gain body condition score, we get some good fertility. Now, we've gone out and looked at a lot of other herds, and this group continues to stay down. This group has maybe come up more. They've come a little closer together on some of these other herds. But this relationship still seems to be there when we're doing, again, the breeding based on these kind of super fertility programs. We can really get a lot of fertility from these high-producing dairy cows when we put in a good program and we have the nutritional management correct. And that's what a lot of herds are doing around the U.S. I mean, if you go to some of these herds that have these phenomenal fertility, that's what they're doing, is putting in a really good protocol and doing a lot on nutrition. So here we go. Decreased fertility in cows with greater body condition score loss during the first 21 days of calving. So that really is affecting somehow something that's going on here seems to really be impacting here. Now, we don't know exactly what that is. People would suggest maybe glucose, with Matt coming up, maybe the NEFA concentrations. There's a lot of possibilities. This kind of study does not show us what the effect is, because when you're having this body condition score loss, there are lots of other things happening. We looked at embryo quality. I won't show you the data right now. We superovulated animals, looked at embryo quality. If they were losing body condition score, actually in that study we had weekly body weights. if they lost body weight, they had poorer embryo quality at day 7 after this AI. So it's essentially very, very consistent with the idea that Jack Britt put out there in 1992, that really body condition score loss is having a major impact. And I know all of you already had bought into that. It was only Milo Wiltbank that had to buy into it, but I think that it definitely is correct. It looks definitely interesting. Now, it's not just body condition, or it's not just energy status that's changing. There's also some changes. There's a low methyl group in those animals losing body condition score. And so that got me thinking, and that's kind of my transition to this next part of the talk about the methionine, because we started thinking, who knows what it is, but it could be, it makes a lot of sense that it might be an effect on the genome of the oocyte, and that's having impacts then later when we're trying to breed that animal. I'll show you there's a really critical part of the genomic effects going on here at this time. All right. So here's methylation of the oocyte. I know you don't really care about this, but what's happening is you're having changes of where methyl groups are being added to the DNA. So methylation of the oocyte comes down, then it comes back up. And it's coming back up right during this critical time period when the animal's going through the negative energy balance. Now, at this time period after ovulation, the embryo also goes through some interesting methylation changes, and you can see the embryonic male genome decreasing in methylation, and the embryonic female genome also decreasing, but a little bit later. Well, methylation ends up affecting how genes are expressed in the body. So the reason your eyeball expresses certain genes and becomes an eyeball as compared to a finger is because of turning on of certain genes. The DNA is no different. And the reason certain genes are turned on is because of some of these effects, like methylation. Methylation is a critical aspect. So if you don't have methylation right, you might get expression of immune cell genes within somewhere else, within the mammary gland or within the eyeball, and those would cause, we call that ectopic expression, and that's a problem. All right, so there's negative energy balance right during this critical time period. So could be effects, low amino acids, low methyl donors. Okay, so now I'm going into methionine. Methionine has anything to do with this. It's, again, this amino acid. It contains the sulfur. The sulfur isn't very important, though, for what I'm talking about. It's encoded by this particular transfer mRNA. It's the start signal for protein, most proteins in mammalian cells. And it can donate this methyl group. So this methyl group is, again, the methyl donor that might be used for these epigenetic effects or epigenomic effects. And what happens? Here's the methyl group, and it gets added to the C of the DNA in these what we call CG islands, and that changes gene expression. I mean, that's how these different tissues then are regulated to express a specific gene, or it can methylate the histones, which are proteins wrapped around the DNA, and that changes gene expression. So that's what epigenetics or epigenomics is all about, is just methylation. And there's different ways to methylate a lot of different compounds. We were, again, using methionine, which has a single methyl group. And this actually stops gene expression. Primarily when you methylate, you turn off gene expression. And then when you're going to turn on that gene, you take away the methyl group. Not always. There's some places, if you methylate, you'll get an increase in gene expression. Certain areas that you methylate. But most of the time, you turn off gene expression. All right, here we go with methionine. Here's the methionine concentrations after fertilization. In the plasma, the oviduct is always a little bit higher. The embryo moves to the uterus and the plasma, and the uterus is a little bit higher. So you've got methionine about double the concentrations in the uterus. What are the requirements? These are studies from Pete Hansen's lab. They're somewhere in that range. They're pretty much, it seems to have plenty for what you need. Well, we went in and did this in vivo, so we did this study, and this is how it was done. Holstein dairy cows, and I'll show you that we then superovulate at the end to be able to look at the embryos, to be able to have enough embryos to be able to evaluate these effects, potential effects of methionine. And we had methionine being fed from calving to 70 days in milk. It was at 2.43% methionine versus 1.89. Lysine was the same, and that's of the metabolizable protein, percent of the metabolizable protein. So within kind of the normal range, but a little bit higher in this methionine group than in the control group. We had an effect on milk protein and protein yield, but here's the milk protein. You can see it's consistently higher in the group that was fed the rumen-protected methionine versus the control. And here's the concentrations of methionine in the plasma, 16.8, kind of on the low end, I think, but that's what they were, and they came up after we fed these animals methionine. So we were having some of the effects we thought. We increased plasma methionine, we increased milk protein concentration, milk protein yield. Alex Sosa came in and flushed these embryos then from these animals. We superovulated them and set them up with or without this methionine. And again, they were all the way from calving out to this time period. We did synchronization, superovulation, because we wanted to have enough embryos to be able to evaluate, is this having an effect? We flushed the embryos. Here's what came out. So we 17.7 CL, no difference in how many superovulations we had, no difference in fertilization, no difference in transferable embryos, no difference in percent degenerate embryos. Essentially, really consistent with the data that Pete Hansen had from in vitro produced embryos, that really by day 7, we didn't, by having lower methionine or higher methionine, we didn't really change the what was happening with our embryo quality. Well, we're very determined at Wisconsin, and so we wanted to evaluate, are those embryos really normal? So Francisco, who's now a faculty member here at University of Florida, did this particular study with these embryos and evaluated the whole transcriptome, every gene that's expressed in these embryos. Is there any difference in the embryos that got methionine versus didn't get methionine? So again, evaluated all of these embryos, or a group of these embryos by RNA sequencing. Here's every gene expressed then in the embryos. Each dot represents a gene. So all of these black dots are the genes that were expressed the same in the embryos with the dams exposed to methionine versus the embryos with the dams exposed not to methionine. And what you see, these are the ones that decreased. Most of the genes that were different, and they're the ones in red here, were in the methionine-treated embryos, and they were lower mostly. There were a few, though, that were higher, interestingly. But most of the ones, and most of the ones that were really a lot, so these are the ones that are expressed in high amounts, and they're a lot lower. This is fold change. Fold change is actually 2 to that fold change. So it's really very, very high, large differences. These are the genes. Of course, you all want to know every gene here. But I'll just show you, for instance, the 2 right here are our immune system genes being expressed in the day 7 embryo at incredibly high levels. This is about 3,000. This log 2 fold change is about 3,000. This is called the false discovery rate. So it's essentially the p-value corrected because we evaluated so many genes. It's very, very likely these are different. I mean, it looks like these are real differences, and we turned off gene expression of immune proteins within these cells. And so there are a lot of different proteins. You can look them all up and see that. And again, I'm sure there's more to come here in Florida with Francisco being here and trying to evaluate and figure out what's going on. So the conclusions from the RNA-seq trial, we've got Methionine supplementation of the dam during early embryo development changes gene expression in the embryo. Most genes are down-regulated by methionine supplementation, which is what you would expect with epigenomic effects. It's still unknown how these changes in gene expression will affect later pregnancy and calf physiology. We have data from other species that there's major effects. Oh, 10 minutes. How did it happen that it's 10 minutes? It's 48. Okay, well, we'll go with it. Okay, and epigenomic effects may occur in embryo when a dam is supplemented with methyl donors. Here's speculations, of course, because that's what I do. And the speculations based on these studies is that differential nutritional balance during the early postpartum period has later impact on the embryo, possible due to oocyte effects. Methionine supplementation of the dam can alter embryo gene expression and possibly later physiological effects, which again has been shown. We should be able to alter the epigenome of the gametes in embryo by nutritional manipulation of the dam. So you ask, does it have physiological or practical impacts? And so I wanted to show you a study we've just completed. We're still analyzing some things about it. That's really the effect of supplementation with rumen-protected methionine, and Mateos Toledo is doing this as his master's. These are the diets, and we just had supplemented smartamine, and the only thing we're changing is this distiller's grain, and you'll see we did this by top dressing. So this is Mateos here top dressing every day for 8 months each of these individual animals, and he either top dressed with a rumen-protected methionine that was mixed in distiller's grain, or the same amount of distiller's grain. So from 28 days to 128 days, he top dressed these animals to try to evaluate again if there was an effect of rumen-protected methionine on some of these reproductive traits. We were trying to get 800 animals. I think we got a couple hundred animals here. I'll show you what happened. But this is the odd thing. When you top dress like this, we get kind of an odd pattern of methionine. We didn't understand this. We didn't know this at first. But you top dress at time 0. Methionine comes up. It's the highest amount and quite high at 12 hours. And then it comes right back down. So that's the pattern. May not be the ideal pattern. May not be the pattern you get if you put it into a TMR. It's the pattern we get when we do this top dressing. We don't get any change in lysine. We don't get any change in histidine. Here's what happened to protein, milk test 3. We didn't get incredible effects. I don't know if this herd was just a little bit on the high side already, but no significant effect here in the primiparous cows, a significant effect in the multiparous cows for percent protein. We came in then again with this double OVSync protocol. The only difference is we put in a second prostaglandin, which improves fertility about 5%. We actually measured progesterone right here, and 5 days later to be able to evaluate if they were synchronized. We only looked at the synchronized cows. Sorry about that. Is that what that is? Okay. We only looked at the synchronized cows, so there were 8% of the cows not synchronized, 92% were synchronized, and the ones that were not synchronized were low fertility. But you can see here, 62%. I mean, that's how these protocols work. They give you kind of incredible fertility among the synchronized cows. OVSync actually does too, only it only synchronized about 50% of the animals. So we're just improving synchronization, but 62%. What happened with methionine? Here we are. So here's the first lactation cows. No effect of the methionine on pregnancy per AI at day 28, day 32, day 47, day 61. But in the older cows, you see this starting to drop. In the control group, a little bit in the RPM. That's because of pregnancy loss. We actually ended up having a significant effect in the older cows on pregnancy loss between 28 and 61 days, or 32 and 61 days. I don't know exactly how to interpret that. Clearly dropping, if this is real, from 20% to 6% is a major impact. I wish we had about 10 times as many animals. It is significant. I wouldn't necessarily tell you to put in rumen-protected methionine to decrease your pregnancy losses, but you know, it's not a negative. It looks pretty good. We're trying to repeat this with larger numbers this year to see if this repeats, but it looks kind of interesting. The other thing that kind of argues for this effect is we measured the embryos. And so we recorded 15 seconds, 2 independent people. We wanted to be able to evaluate it independently of knowing treatment group, etc. We just measured the embryo. Here's the embryo. We measured crown rump length, abdominal diameter. We also measured the amniotic vesicle, longitudinal transversal. And kind of interestingly, this is how it came out. Overall, we had some effects here. But then when we looked at parity effects, so here's the primiparous cows, not much of an effect. So again, here's amniotic vesicle in the longitudinal region, no effect of control versus RPM. But in the older cows, a significant effect. And you can see across there significant, tendency significant. So it looks like we're growing the embryos a little bit larger with the methionine. Is that an effect of the epigenomic? I don't know. Is it an effect of the protein? Hard to say. Is this better? We don't even know that for sure, but it seems like it's related to that pregnancy loss. So we come back to the end. I talked about this transition period with body condition loss. Loss of body condition early, postpartum, can reduce fertility. I think, again, most of you believe that, and it seems to have shown up pretty clearly in a pretty big study. Epigenomic effects you can get on the embryo. And we could get it by methionine supplementation. Maybe this reduces pregnancy loss. The big thing is, I think in the future, you're going to be starting to feed that dam a little bit differently to try to be able to change the characteristics of that calf. At some point, as we start to understand this more. Right now, we're right kind of on the edge trying to figure this out. But I think epigenomic effects are going to have some impact on nutritional programs.